Hi everyone, today we're going to be studying Chapter 9, which is talking about controlling microbial growth in vivo and the use of antimicrobial agents. So in this lecture we're going to discuss the characteristics that make up the ideal antimicrobial agent. We'll discuss the attack mechanisms of antimicrobials. We'll also look at some antibacterial agents, antifungal, antiprotozoal, antiviral agents. All of these agents have been specialized to target the various different types of microorganisms, which we've discussed extensively in this class. We'll also touch on the concept of drug resistance, and then we'll discuss some strategies that are used to defeat drug resistance. We'll talk about some empiric therapies that exist, and we'll talk about the effects of antimicrobial agents or the side effects of them. And then that'll be it for this particular unit. So to start things off, let's go ahead and define some terms. The first term I want to define is that of chemotherapy, which is essentially the use of any chemical agent that's going to be used to treat any disease or condition. So when we talk about the root word chem, we're referencing chemical, so that's how we get chemotherapy. And therapy, this STEM word is going to be synonymous with the idea of treatment. So chemotherapy is treatment through the use of a chemical, which we know in the form of drugs. A chemotherapeutic agent is any drug or any treatment used to treat a condition or a disease. So chemotherapeutics are what we use in the process of chemotherapy. Now, there's all kinds of different microorganisms out there that can elicit infection, right? So anything that is typically microscopic that induces infection can be killed by what's called an antimicrobial agent, which also is a drug that is used to treat specific illnesses. And the way the antimicrobials do this is they can inhibit or they can kill a pathogenic organism in the body, which is what in vivo means. Now, antimicrobial agents, for the most part, can be considered to be antibiotics, but we do have some antimicrobials out there that aren't antibiotics. So something like a hand sanitizer is fitted with chemicals that are used as antimicrobials, but we don't consider the elements in hand sanitizers to be antibiotics because we don't take those in vivo, right? So instead, those are very topical kind of agents. So some antimicrobial agents are antibiotics and some are not. Some other terms to be familiar with are antibacterial agents. These are going to be specialized chemicals or specialized drugs that are used to treat only bacterial infections or bacterial diseases. And then we have antifungal agents, which will not have any effect on bacterial diseases, but instead they will target and treat fungal diseases. And then next we have the antiprotozoal agents, which targets diseases that are caused by protists. And then we have antiviral agents that target diseases caused by viruses. And an antibiotic is any substance that's used to either kill or inhibit the growth of another microorganism. And typically these are going to be things that you put inside of your body. So antibiotics are used to kill and inhibit the growth of microorganisms. In this diagram here, we're kind of looking at the complexity that's involved in antimicrobial therapies. So when we treat micro-infections caused by microorganisms, we've got to use an antimicrobic drug, which can be either a drug that has been synthesized from an organism or a drug that has been synthesized from chemicals in a laboratory. So if you have a drug that's been produced using elements from another living organism, we refer to that as an antibiotic. If you have a drug in which you take components and manipulate it inside of a laboratory setting, you refer to that as a synthetic drug because it's been synthesized or made by people. So there's side effects that can happen whenever we put different drugs inside of our body. You might have a non-reaction from the host, so the host body might reject the drug treatment, and maybe just that drug just passes straight through the system of the host, and there's no residual drug, so no treatment plan is actually able to happen. Sometimes you can have drug delivery problems. It kind of depends on how the drug has to get into the human body in order to work. If it's a pill that you're taking, sometimes people might be allergic, or sometimes people have a gag reflex, but they don't want to take their body will not be able to take the drug, so that way treatment can take its course. If you have to do an injection using intravenous or intramuscular injections to deliver the drug into the system, that can be quite traumatizing for children. It's also a very invasive procedure for adults. So sometimes you might have some problems if you have to do an injection with a drug. Some drugs can be topical. The tissue in which you're applying the drug to may not allow the drug treatment to pass through the tissue, and so the uptake in the body is not very efficient. Of course, you can also have side effects when it comes to the use of antibiotics. The toxic nature of the chemical being added to the body can cause problems or complications to the patient. The patient might be allergic to the drug treatment. You may also wipe out a number of microorganisms that are part of your normal flora. And remember, your normal flora are all the bacterial organisms that reside in your body. And so when you disrupt that flora, that leaves your body subject to having a high risk of being colonized by more deadly bacteria or more dangerous bacteria. Sometimes a doctor might diagnose you or might prescribe to you the wrong treatment option, and as a result, you might have drug delivery problems as well as side effects, but at the same time, you don't have any activity because it's the wrong treatment plan given for you. And sometimes this can be complicated because microorganisms are incredibly microscopic, and when they infect your body, sometimes the symptoms are very similar to each other. So in some cases, you may be suffering from an infection caused by a virus, but you might be receiving treatment from an antibiotic that targets a bacteria. And so if you prescribe the wrong drug treatments, that virus is going to run rampant because it's not going to respond to the therapy that was prescribed to you. So that can be another potential issue that you may encounter. The other big issue is that of resistance, where you have bacteria organisms resisting or evading the drug treatment that has been ensued. And this can be due to different mutations that have taken place. It can be due to the passing of the resistance gene between bacterial cells. So drug resistance is a pretty bad thing because it enables the pathogen to become stronger, and the pathogen has been able to evade the drug treatment that's been prescribed. In order to prevent resistance from happening, you need to make sure that you take your medications as prescribed and to complete the treatment plan. Sometimes when we start feeling better after undergoing our treatment plans that have been prescribed to us, we don't finish our medications, and because of that, not all of the bacteria that were harming our body have been depleted from our system. So you have some survivors, and then those survivors go on, they can pass on these resistance genes. If you take high doses of medication for a long period of time, or if you take medications in combination, that could all lead to helping to minimize the chance of resistance developing in certain organisms. There's different toxins that are released in the body whenever you take a treatment regime. So if you're treating bacteria that are gram-negative, you may have a drug that kills the bacteria, but when that bacteria are killed and they are lysed inside of your body, what happens is you have the release of these toxins, endotoxins primarily, and when those endotoxins are released, they're released in the form of lipid A, which remember is a component that's included or embedded inside the gram-negative bacterial cell wall. And that can be very toxic. So in the sense, when you kill these bacterial cells in the body, you're actually kind of opening this Pandora box of toxins, and those toxins can cause a lot of necrosis or a lot of harmful effects inside the body. So those are some side effects that you have when using different type of treatment options. But our primary reason for using these obviously is to prevent the growth of bacterial organisms. We have bactericidal agents as well as bacteriostatic, and bactericidal means that you kill all the organisms that are causing infection, and bacteriostatic mean you simply inhibit their growth, so that way you have smaller quantities of the bacteria that's infectious to your body. And then some drug treatments can also enhance your immune system, so this puts up or enhances your host defenses, and this helps to remove pathogens from the body. Our biggest kind of bullet that we use to treat a lot of gram-positive infections is penicillin, which many of you know was discovered by Alexander Fleming, pretty much on accident. Fleming basically left out some petri dishes, and he saw that fungus, which is called penicillin, it's a specific type of fungus, was growing on the agar plates, and where it grew, you could see this zone of inhibition in which the penicillin would not allow bacteria to grow around it. So there was kind of like this halo, or this area of no growth called a zone of inhibition that prevented bacteria from growing in that spot. On this particular plate, you can see colonies of Staphylococcus aureus, which are all these little colonies all over. You can also see a penicillin colony, which is a fungus. And this fungus right here, the picture's not very good because it's black and white, but you can tell it's penicillin because it's larger of the bacterial colonies. And so this penicillin has released something to inhibit the growth of all of these bacterial colonies in this B region here. So this area is very minimal in terms of bacterial growth, and you can see that there are some colonies in it that have been struggling to grow, but they haven't been able to do it because the penicillin is putting out enzymes that are causing the bacterial cells not to grow. And so from Fleming's observations, we were able to isolate this penicillin and package it in the form of pills or in the form of liquids, and be able to use that to treat bacterial infections in the body. Okay, so what makes the ideal antimicrobial agent? Well, first off, we want to make sure that it works. So it needs to be able to kill or inhibit the growth of the pathogen. Secondly, we also want to make sure that it doesn't induce more harm to the host that's consuming the antimicrobial or using the antimicrobial than what the disease-causing agent is doing. We also want to make sure that there is no allergic reactions that take place that kind of coincides with causes no damage to the host. But allergic reactions can be very severe because you never know how people will react to them. Sometimes you have a type 1 reaction, sometimes you have a type 4 reaction. The more severe the reaction, the more problematic it's going to be for the patient. Some other things about ideal antimicrobial agents is that we want to make sure that they are stable when they're stored. They can last a long time. We want to make sure that they are able to reside in the body long enough to be effective and to kill pathogens before they mutate and become resistant to the antibiotic or antimicrobial agent.